today we'll be looking at the topic CNN from scratch. Obviously, you have no idea about what is CNN. Maybe you would be knowing its full form. That is convolutional neural network. So this convolutional neural network, so there are different types of neural network. Okay, the different types of neural network, just know the name. That is ANN, artificial neural network, convolutional neural network, maybe graph neural network. Okay, so there are different types of neural networks. So when we talk about convolutional neural network, most of the times it is used for image, like for computer vision. Okay, to process images and learning with the help of images. So you can write this as computer vision. So this is the third day of phase one, where we'll be trying to strengthen our foundation. So on this first day, we are starting CNN from scratch, and let us see why the CNN is right tool for histopathology image classification. We'll tell you the reason, but the main reason is that it is a very rich type of image. You know, the color channels that are produced, the patterns that we have to detect are very rich. CNN will be quite helpful to detect all these attributes. And then let us also, we'll see that how image is represented as a grid of numbers. So I told you if you have an image, so the image will be divided into pixels, and each pixels will have some color and its intensity value, the number 0 to 255. So the images can be represented as number for the computers to understand. Next, we will understand what actually is convolution and what is activation pooling. So these are some of the terms that you must know. So convolution, activation, pooling, and we won't be doing the math, but maybe we'll try to understand that how, with the help of math, CNN is happening. I've already mentioned that any neural networks, they have different layers, and in each layer, they will try to learn different aspects of whatever task you have given. So in CNN, in different layer, it will try to learn different pattern of the images. And usually each layer will be stacked on top of each other. Okay. So as the new layers are stacked, the model is learning something else, something more. In the first layer, very simple pattern it will learn. And suddenly, as the layers are stacked, the computer model will be trying to learn increasingly complex biological features. And we have already discussed this in the last class. Like, obviously, we'll look at the color, we'll look at the density, we'll look at the shape of the glands. Okay, we'll also look for any mitotic figures. Okay, mitotic figures, division. So we have already, what are those features? We have seen that in day two when we were diving deep into, about what are the basics of histopathological images. So that computer will try to learn. Then there are something to learn through different layers. There are something known as loss function, backpropagation, which is very important for you to understand first, okay? Then definitely transfer learning, which is not actually, a core part of CNN, but since we have used it in our project, we'll be having a glance at what is transfer learning. And the architecture that we had used is efficient net B0, which in turn is a part of a larger architecture that is ImageNet. If you're not understanding this particular part, when the time comes, I'll explain it in detail. Okay. And then we'll also try to understand how to connect this computational part, that is CNN, back to pathologist level reasoning, who are totally, biology person. Okay. A person who doesn't understand coding or doesn't understand, computers, will try to how they can connect, like how they can use our model. All these things also we can see. Then apart from, when you learn the model, the final output, what you need to generate is how many percentage, accuracy you've got, 95%, 75%. But I'll tell you one thing, that just getting this mathematical numbers accuracy percentage is not enough. Okay. There can be a fair chance that you will have higher accuracy percentage, but it might have not learned anything important, but which is, which has a biological importance. Maybe the image was dark enough. I think Harry Om had a doubt, like he could not understand what was the difference between the two images placed next to each other, because the benign one also showed some purple round shape, and the malignant one also showed some purple round shape. So if even the shapes are a little bit similar, there are some other aspects I have taught you that you see. You can see which color dominates, mitotic figures are there or not. Then we'll see what is the density, and then we will see, you know, the pattern is clear or it is very random pattern. So there are so many things apart from, gland shapes that we can see. Suppose you have built a model, and after testing your model, you're getting a 99% accuracy, but in the real life, it is not considering all these patterns. Okay, if it is seeing a circular gland, it means that, oh, okay, it is benign. And if it is not seeing any circular gland, only then it is malignant. So, no, there are different grades of cancer also. Sometimes malignant, it takes time to transition from being malignant, so from being benign to malignant. So in between, there are several grades of cancer. So, when a benign tumor is being transitioned into malignant tumor, the normal features are not lost suddenly overnight. Okay, they are not lost suddenly. So it takes time. So at that time, what happens? Your computer model can misclassify. Okay, misclassify your model, and it will still give you a higher accuracy percentage. And that is the reason I'm not straightly jumping into coding. Okay, this is how you make layers, and this is it. I can teach you within, I can have a 5 hours workshop and I can teach you everything, but you'll not understand, and your model will never work. Understand biology and make the computer to understand that biology as well. So just getting a higher accuracy percent is not enough. Okay. So you definitely are aware of what problem we are solving. Mostly these introductions are redundant, but then also, just let's have a look. Like suppose a patient comes, she has a lump in her breast. Okay. So every time a lump does not mean that you are suffering from cancer. It may mean that maybe a normal cell of extra growth, or it may be a benign phase, or it may be a malignant. It can be anything, right? So what you do, you cut a tiny piece of tissue and you send for biopsy, and under the microscope, you prepare those slides and you take the photo. Okay. Now, if you're very skilled pathologist, then what you can do is you can see and you can understand what type of tissue it is and what grade of cancer that is, and you can plan your treatment entirely. Okay, but the amount of trained pathologists, especially in the third world countries, is very less. Okay, and even in the developed countries, if you have skilled pathologists, the demand is so high that even there, the patients are on waiting list. So what if we provide them such a model that just, you have to take the pictures of the histopathological slides and feed it into your computer, and you can get a classification with certain percentage of accuracy that this uh, belongs to this particular type of tumor. Okay, and it can also tell you what is the grade of the tumor, right? So it will save a lot of time. And most of the people can get access to the expert pathologist because it is saving some time for him. Okay. Let us move on and let us see what exactly does the machine needs to decide in this case. So first, in the first level, suppose you see level wise, it must decide whether it is cancerous or non-cancerous. It is just a tumor, just an excessive growth, then it will be benign. So, and the second, maybe if it is cancerous and dangerous, then it will be classified as malignant. So, on the first level, your computer needs to decide whether it is benign or malignant. And after that, I have, opened the folders of the data sets in the last class, and I've showed you. In the benign folder, we had uh, different types of we had different subtypes. And under malignant folder also, you had 4 different subtypes. Now, first, if the computer had decided or it had learned that a particular image belongs to benign type, it will move on to the next level and it will try to subclassify it. If it is cancer, if it is malignant, okay, which type of cancer? Is it, serous, it's present in your serum or sera, okay, mucinous in mucus layer, or it is invasive ductal or lobular. So these are type of the cancers that uh, are provided in our data set. Okay, so don't worry, it will be provided in our data set. And that is why most of you, maybe because you are in the beginner level, you should understand whatever work you're doing, not only AI, not only ML. So any bioinformatics work, if you're doing, your data set must be well labeled. Each folder, each image, each file should be well labeled, and the names of those files and folders only will contain this, uh, uh, you know, um, this information. Because inside the serous folder, in mucinous folder, or a ductal folder, a lobular folder, different types of images will be arranged. So from that, it will extract the name. And we had also seen that we also had metadata file. So what is metadata? Data about the data. So we had some CSV, separate CSV files. Like these are nothing but a kind of spreadsheets, like Excel spreadsheets, comma separated value files you have. Okay, in that also your metadata would be written. So not only the images, we also need to give well labeled data. Okay, and we should be, apart from well labeled data, you can also use the extra files that has been provided. Now you have to note one thing down very clearly. When you already have the names of the different types, and you should only categorize your data. So you have all the images. Can I get some voice from your side? I've shown you the 2 data sets, that is break his and UBC ovarian cancer set. Yes,
Yes,
you know,
ma'am.
we're working
Yes,
with
ma'am.
2 data set, right? And each data set, when I had opened your folder and I'd shown you, each folder, each file had names written over there. So names are there, well labeled. It will extract some information with the help of names as well. Without names, how will you be able to categorize all this into benign, into benign or malignant? And if suppose it has been diagnosed as benign and malignant, then you have subfolders with the subtype names, and then it will try to classify. Another level is another classification, right? Now the grading work was not done in the previous initial phase. Let us see what is grading. So now you have understood that maybe the malignant, it is malignant, but some sort of glands are still present. That means it has not reached its final stage yet. So the final stage has not been reached yet. So we can grade the cancer level as well. We'll try to do that as well. Okay. So these 3 tasks can be performed. And I told you, so basically this is a categorizing task, categorization. So in the world of machine learning and AI, when you have names, please note it down. Okay. The first thing, your data is well labeled. That means you have name, and you just need to categorize them. Okay. You have to split it into certain categories. Now, this type of work is known as classification. Someone, have you heard about it? So basically in the world of machine learning and AI, there are two types of tasks. The first one is classification task.
Classification,
Yes, because
regression.
you have a well-labeled data, every image, and even if you're not working with image, if you're working with any file, you are having that data well labeled, right? You just need to classify. So already there's some supervision, okay? Something is helping you, something is guiding you, there's some supervision. But this second type of task, suppose you don't have any type of, you have got the data, right? But your data is not labeled. It doesn't have name. For example, you just have some random pictures of dogs and some random pictures of cats, and each image is not named, okay? It is not named. And still, if you're telling to group the pictures of dogs and cats separately, and the data is not labeled, then it will be known as clustering. So you should repeat in your mind, or you can repeat it right now. There are two types of tasks. If you have labels, it's classification tasks. If you don't have label, it is clustering tasks. Okay, but we are going to, because our folder is well labeled. Every image file is well, nicely named and organized into labeled folders as well. So we have a classification task, and that's why the name of the project itself is classification of tumor subtype. Understood? Did you get this concept? It is an important concept. Did you?
Yes,
Yes, ma'am.
ma'am.
Yeah, because why classification? Name is already present. You are working under some kind of supervision. You are getting some kind of help because the name itself is, subjective and it's telling you something, right? Next, if classification is known as supervised machine learning or supervised deep learning, whatever, supervised task, okay, supervised ML, supervised deep learning, supervised neural networks. So if it is a supervised task, this will be known as unsupervised. Okay, understood? So this is some basic. So I didn't took this topic in the basic, separate as a basic class because I didn't want to waste time. See, I hope you have understood now. So this is the classification task that we'll be doing. First, we'll do binary classification, and then we'll jump into the subfolders and we'll try to categorize them as different subtypes. And if possible, and if our model is working very, efficiently, we can also try to grade at which, whether the cancer has advanced or not by just looking at the different patterns. Okay. And that is why I've told you to please go through the day 2 of video again to understand the biology behind the histopathological images. I will be uploading it by tomorrow. So what is the big intuition? Never forget this. Your eyes look at a biopsy slide, and what does your brain say? It says that, okay, you can see some abnormality. Okay, it's abnormal. In fact, a CNN will try to do the same thing, except instead of neurons, it is using different layers of mathematics. Every concept, okay, it is an analogy, okay? It's like an analogy, like it maps directly onto what your brain does will try to make the computer do the same. So how the humans see images, let us have an intuition about it first. So our eyes, so it's like a class 10 topic. It will capture light. So what analogy? Like it's a camera sensor. So a retina, where the image is forming, it will detect the edges, it will detect the colors, it will detect the texture, like um, our rods and cone cells. You remember? Our retina has two types of cells, rods and cones. Cones are responsible for color, RGB. Rods are responsible for the intensity, brightness, contrast. Okay. Now, when these signals reach our brain, it will combine all the different patterns, different edges into shapes. And then we can understand, okay, this part, it is dark in color and it is circular, it is nuclei. It is dark in color, it is not circular, it can be nuclei, but it can belong to a malignant image. Glands are like rings-like structure. Okay, they are arranged in a ring-like structure, right? Next, step 4, your frontal cortex. Now you have understood the image. You have to do reasoning, okay? So the reasoning part is done by your frontal cortex. Frontal cortex is not fully developed until the age of year 25. Please understand this. So you cannot make much wiser decision until you reach the age of 25. Just an information I wanted to give. So how the CNN works, like how this can be mapped into computer level work. So similarly, like a camera sensor, the computer will try to capture each individual pixels, and each pixels has a range of 0 to 225. 225 is the most vibrant, brightest, or the most depth of the color. Okay, now just a second. Yeah, so now after the pixels are detected, different layers will be responsible for detecting different patterns like edges, colors, and other transitions, those things. After certain layers, when we deep dive into different layers, after it has learned some basic parameters, they will try to, in certain layers now, complexity arises after some layers. And then they'll try to combine each and every parameter. And then they'll try to study the shape and structure. And then there is a function known as, after all the layers, after all these things, many things happens over here. I'll try to tell you today itself. But let us see what is the output. Output is nothing but, after studying all this, after learning the image, it will just try to give you probability. What is the probability of this particular image to be malignant or to be a benign one? So this is just a, you'll never get 100% result. If you're getting 100% result, it might be, there's also a term known as overfitting. So what do you mean by overfitting? Anyone? Can you tell? Anyone? Can just by the name, can anyone try to guess what do you mean by overfitting?
Ma'am, overfitting happens when model is too complex.
In the model, you're telling that when the model is too complex. No, overfitting can also be done when the model is very simple. Sometimes when you have to screw or stitch something. Yeah.
Also said that when training data include very much noise, then chance of overfitting.
Let me put it this way. Now let me explain it to you. Suppose you have to stitch a cloth. You have to sew something. Okay. What will you try to use? Will you use a needle or will you use a sword? Which one will you use?
Needle.
Yeah, at the time, suppose your model is giving something equivalent to sword, that is overfitting. You understand? Sometimes a simple things can get the work done better rather than, making a bigger thing, Just imagine if you're using sword for sewing, you can never do the work. And a very simple example, suppose you are preparing for any exam. First one is a rote method where you're mugging up everything, okay, without understanding. You might get 95%, but it will not, you have not learned anything. Nothing you have learned. This is overfitting. Overfitting is just memorizing the patterns, and when at this time if there's overfitting and you're getting maximum probability your model is giving this accuracy, but when you're feeding it with the new patient data, this model will fail because it has never learned, it has only memorized. I hope you understood. But if you have learned the real pattern, okay, if you have learned something well, even if you have got 75% accuracy, still that model can perform in new data as well. I hope everybody you understood the concept of overfitting.
Yes, ma'am.
Yeah,
Yes, ma'am.
that's all. Okay. So we don't want that. We'd never go behind this 90% or 100%. You should see whether my um, model is learning meaningful data on. Next. So the core insight. A CNN, convolutional neural network, is a mathematical model. Okay. Just like how our visual cortex works, it will try to mimic the exact process how the brain use. Okay. But just that it will use numbers, and it will use mathematical data structures like matrices or tensors. You don't know what are tensors, I'll tell you maybe someday else, instead of actual neurons. Okay. I hope till now you have understood what is an image mathematically. So each image you have pixels. If it is a black and white image, so black and white image, you have each pixel you have 0 to 225, 0 being black and 225 being white, and all the different shades of gray lying in between. And if you have a color image, each pixel will be divided into 3 channels. Okay. So that's why a grayscale image will use less computational infrastructure as compared to a color image. So more color images are more complex mathematically because each pixels has 3 dimensions: red value, green value, and blue value. Okay. You can see like 3 dimensions. Understood?
Yes.
So H&E staining, hematoxylin stain, nuclei is dark blue again. So at that time, if it's dark blue, that particular pixel value at the blue channel will be more. And wherever the eosin stains, suppose like cytoplasm and stroma is nothing but the surrounding regions of the glands. So that it will be pink. So blue values will be less and reds will be more. Okay. So machine will try to read these channels and see if it is more blue, that means negatively charged particle like nuclei or DNA, they are stained. Okay. And if it is not stained blue and it is pink, that means it might be stroma or it might be cytoplasm. Okay, and that's how the programming will learn how the colors are encoding the biology. Okay. Now, another thing that I want to tell you is the huge complexity of the histopathological data. Now, the whole slide, this image, so like length and breadth, okay? So the resolution, it's very high resolution. So you have got like 100,000 by 100,000 pixel if you have got. So how many pixels per slide? You have got 10 billion pixels, which is a very huge number. Okay. Right? Can you see? That is why histopathological images are very high scale. And now a whole slide has 10 billion pixels. Now each pixels has 3 channels. You see? Again, much more higher dimensions. So the 10 billion numbers corresponding to each pixels, since it has 3 channels, now becomes 30 billion values per slide. So it is very huge. Okay, the amount of heat generated, the amount of computations needed, the amount of money you have to spend is very huge. So what we do is the whole slide we divide into small overlapping, might not be overlapping, like adjacent patches. It depends whether it is overlapping or not. It depends on choice. Okay, small patches. And at a time, we will learn a single patch. And that patch size is only 224 by 224. This is 1 pixel, 224. Sorry, not 1 pixel. So it has 224 by 224 pixels. And multiplied by 3 RGB channels, now you have got 150,000 near values, which is much more manageable as compared to 10 billion numbers. Okay. So why do we use these patches? CNN input must be of fixed size. Now, every CNN model, it requires a particular fixed size image, random image you cannot give. That is why you have to pre-process your images, which will be doing via Python by coding. So this patches, it won't give the whole slide at once, but smaller area, local context, first it will learn. Okay. So basically a huge task is being broken down into smaller, simpler tasks. Understood? Many smaller, simpler tasks. I hope this is clear. Till now it is clear.
Yes.
Now let us start with the convolution step. It is the heart of CNN. CNN, what it does, it learns this. The purpose of this neural network is to learn which part of an image to pay attention to. So it won't take the whole image at once, never. You need such a powerful graphical processing units for, image processing. What CNN does is like it only looks at certain parts of the image. Let me tell you one very basic thing. Okay. What happens, suppose if you have got a data set, okay, there's so many different information. So in data set, so many different informations you're getting. For example, if I give you a different data, suppose you have got, a protein sequence file, protein 1, and you have got second protein 2, you have got third, protein 3, protein 4, like that. Suppose you have got n number of protein files by informatics workflow in proteomics. And then you can see, different properties like this protein, what quantity it has been produced, what is the, effect of the protein. So you have certain rows and then you have got certain columns. So the columns, like quantity, effect, these are known as features. Okay. These are known as features. Now, suppose you have this data set for each protein P1, P2, P3, P4. There are 100 features. What machine learning, what deep learning or AI, what it does, it won't compare all the 100 features to learn something. No. It will pick those features which are most variable. Okay. For example, if I tell you, just 1 second. There is a person A, and then there is a person B, and then there is a person C. If you want to know about these people, okay, now for A, the first feature is their name. Second feature can be their address. Third feature can be father's name. Fourth feature can be school. Fifth feature can be their interest. Have you followed till now? Everyone?
Yes, ma'am. Yes, ma'am.
Yeah, so there are different features. Now suppose a person wants to learn Suppose they are prompting some questions, and if you want to, a machine wants to learn whether this particular person, it has to, classify, or it has to predict whether it is a person A, person B, or person C. Now I'm giving you one condition. They are siblings, okay? They are siblings. So their address might be same, father's name will be same, mother's name will be same. So can the computer learn from these features that they are different people? If they have same address, same father name, same mother name, tell me,
No,
they
it
cannot
will only
use.
look for the name.
Yeah. So it won't give any differences, right? So what is the most variable feature it has to search? The most variable might be name, might be their age. Age is better. Why? Because it's a number also, right? Their interest, their hobbies, which school they go. If they go to similar school, then again it is useless. So any model, what it tries to does, it learns by itself. You don't need to give the variable feature. It will find out by itself. So the most variable feature, it will rely on variability. Okay, it has to learn. It has to learn from only variable features. And even if suppose you have got 52 variable features out of 100, even learning from 52 variable features for 3 people, so it becomes 52 into 3 features, 156 features it becomes. So it is suppose it's a large number. So out of these variable feature, what it will do is it will try to pick the top 10, top 5 variable features like that. Now if it has picked top 10 variable features, now it has only to learn 30 variable features. So like that, this process is known as dimensionality reduction. The more number of features, it is higher the dimension. Now what you did, you rejected those features which are similar. You're not including it for learning. Even if you had got topmost variable features, even with that also you're filtering, and it is taking the top 2, top 3, top 5 variable features. And this process is known as dimensionality reduction. Very important, because bioinformatics data is huge. We are working on big data, right? Similarly, instead of looking at the whole image, it will only look for the most variable parts of the image. Which part of the image to pay attention to, what to look for in those parts. Okay, and all these things will be done automatically. Understand? You do not program the CNN to look for nuclei. You just show it thousands of labeled images. That's it. Just load the images. You only need to know how to load that particular image folder with the help of coding. The CNN will figure it out by own. It will try to learn layer by layer by its own. It will look for irregularities. It will look for larger patches. It will look for darker patches. And if they have got most of these patterns, then it will correlate it with the malignancy. If regular patterns are visible, if smaller, finer particles are visible, and it's not too dark, mostly pink, less blue, then it will be benign. It will try to learn by itself. Okay, got it? So there's a
Yes,
flashlight
ma'am.
analogy for the CNN. So how the CNN works. So imagine you are in a dark room, and you have got a flashlight with you. Okay, so looking at the whole image is like, if there's electricity supply, if it's on, the light is switched on, you're looking at the whole room. But CNN analogy is like that. It is a dark room, And with the help of a flashlight, you are seeing only certain patches or certain area of your room. Okay? And you are looking for only the object that is looking for your phone, rectangular in shape or a specific color. Just give me 1 second. I'll be back. Yes. You got it? Why would you see for circular things? Will you? If you're looking for your phone, will you look for triangular things? No. You know the color of your phone, you'll be looking for that color itself. If it's black, you'll look for object that's black, rectangular most probably. Yes. And you'll move systematically across the room, not randomly. You know where to search. If you know the place, it's well and good. You'll go to that particular portion of the room and you will search systematically. Okay, and each time you shine that flashlight, looking for a specific shape. So convolution is this process, just like that. Okay, smaller patches it will look for. Have you understood the real life analogy till now? Yes.
Yes, ma'am.
How in real life you are looking for your phone with the help of flashlight in a dark room. Okay. Mathematically, how it has been done. Same thing is done, you see. Now I'll tell you, can you see this here in the diagram you see, okay? So this is a image patch of 5 by 5 pixels. So from this whole slide, okay, you have got, okay, let me just color code it. So it's like whole slides, you have got pinks, okay, and you have got some purples. I hope the purple is visible, so whatever. So what you do is, in a convolutional neural network, only a small patch is being worked upon. This patch, if I magnify it like this, okay, so it is 5 by 5 pixels. You can see over here. Coming back to the main image here. So this is 1, 2, 3, 4, 5, and 1, 2, 3, 4. Okay. So consider it like a 5 by 5 pixels. Okay. It's a 5 by 5 pixel you have taken. Now, instead of a flashlight, what happens is you are passing a filter. Apart from a flashlight, you're passing a filter and you're setting some size. So in the case of flashlight, this is your torch or the flashlight over here. So the light rays, so the beam of light, the area of the illumination is this much. So if you have a smaller torch, the area of illumination will be small. If you have a larger torch, the area of illumination will be larger. Got it? So just like that, there is something known as filter, or also called kernel. It will slide over the image. Definitely it will slide at an entire image, but this entire image, it will pass patch by patch. Okay, so one patch at a time. Now suppose you have got a patch which is this big. What you do, you set a filter, you set a determinus filter size. Most probably we are using 3 by 3, 3 pixels by 3 pixels. So what does this do? It will start hovering. That means it will start moving over your image patch. Try to understand. Your filter, like a flashlight, anyone, you can stop me and you can ask me again, okay? So is a filter. Suppose you have decided my filter will be size 3 pixels, 1, 2, 3 pixels by 3 pixels. So this big, okay? 3 pixels by 3 pixels. It will be placed over that image patch and then you will slide it over one by one. So the computer will learn the pattern wherever the filter is at this point. Understood? Let us look at what is written over here. So it is nothing but like a sliding window. It will slide over your image. This filter, it will slide step by step. It slides across the entire image. It will scan every region, every pixel, and it will ask, does this pattern match with what I'm looking for? Okay. So this is the way the scanning of the images is done by convolutional neural network. Okay. So this here was the image patch, and sorry, this one was the image patch. Okay. And filters are sliding. So in each patch, what it will does, it will map it, which part is important. Okay. So this area was important, it will retain it. This particular area was important. So here the features what are present are important. Here are important. So what it does, okay, the unimportant regions are discarded. So you can see many empty regions over here, right? So this is known as, it will generate a feature map. Remember this word. Now while coding, no, you'll be coming across these words again. So I hope you understand what are these. We have feature generation is a process. You have to generate this feature. Okay. So feature map is generated. Again, repeating. You have your image. Instead of whole image, we are taking a patch of image. On the patch of image, we are sliding a filter. Okay. Filter will look for important regions or important feature. So not important areas are discarded and a feature map is generated. Got it? Is this easy now?
Yes, ma'am.
Yes. What is the map? Definitely you'll not be understanding, but I'll try to, you know. So there are different types of features that they have to learn, you see, in different layers. So the first feature, suppose you are detecting where are the edges. What do you mean by edges? Suppose this is a slide. Here it is a gland. So you can see the red boundary. It is the edge of a particular gland. Suppose here you have got some, irregular edges like this. So this one is benign. This one might be malignant. So like that, first it will look for how the edges are, whether it is round or whether it is irregular. So, but everything which it wants to learn, no, let us start with a very simple step of learning. Apart from looking for curved or round edges earlier, it will start by learning, let us look for any vertical edges, because vertical edges, that means it is a straight line. So it will learn whether are there some straight lines or not. Then it will go for some more complex tasks, whether there are, irregular edges or circular edges or something like that. So this is like a, scanning of the image for edge. Now, please try to understand what I'm trying to tell you, how it will detect an edge in image, okay? I'm trying to draw a diagram over here. This is my image and I have one pattern. Suppose some gland, and this is the pattern. So this area is roughly vertical. So how it will understand that this black color, okay, if I zoom in right here, just a second, let me zoom it. Okay, fine, zoom. Yeah. So this area, this black color, your brain can understand that this is the edge of some shape or some feature. Yes or no? But how will your computer understand? What it will do, here there is white region, and on the right side also it has got some white region, and in between it has a, black pixelated region. Yes or no? In between there are some black pixels as well. Yes.
Yes.
So just like that, if this particular image patch over here, okay, you see when the filter is sliding across the image, Out of this 5 by 5, first column and second column, they have very low value, pixel value, 10, almost corresponding to black. Okay. And the third column, fourth column, and the fifth column, they are having higher value, 200. 200, that means they are most towards like white, or in your case, like pink or whatever, like higher intensity. So this might be the edge. Which one? Where you have got the transition from dark to white. So this is the edge, yes or no? Where the dark part is finishing and white part is beginning. Or when the white part is beginning and dark part is finishing. Definitely that will be edge, right? So if you look at
Yes.
this image, don't look at inside whatever it is. Look at this dark rectangle. You know, when the white is ended and black is starting, so this is the edge. This is the edge. Do you get it? Do you
Yes,
get
ma'am.
it?
Yes.
Yes.
Yes, ma'am.
Yes.
Yes.
So what happens now is, just a second, okay? बैठ जाओ हाँ दिन भर यही देख रहा बैठ जाओ मैं क्लास ले रही हूँ yeah, you got it. Now let us understand how this filter is working. There's one. So this is the map. Let us not look at the map. So this all this thing, you know, sliding of the filter corresponds to some mathematical equation only. Sliding of the filter is some mathematical equation. You should get a high value after solving whatever is this. Okay. I just forgot, pardon me. Someone with the math background, can I get any response from them? Let me ask you, let me have a conversation with you. Someone, no?
Yes, ma'am.
Harsh, I guess.
Hariom.
Hariom, you have math background? Have you learned matrices and determinants?
Yes, ma'am.
Yeah. So can you see, now this is a basic matrix multiplication and addition over here. Okay. Just a second. Yeah. So when you see, if others, if you don't understand also, try to just follow. Doesn't matter, it won't be much useful, but if you understand, it's well and good. Now, can you see these filters? This filter doesn't have any pixel value 0 to 225, 255. No, it is not having those values. Your image patch, definitely it is having the pixel value. The black means 0 and white means, or the highest color means 255, right? So here it is a very low value. It is a dark region or black region. Here it is a very low value. It is a black region. So here in the three columns, they are higher number. That means they are brighter region, more white region, right? But your filter, you're assigning some numbers. Okay. So in mathematics, you have got something like matrix. Matrix is arrangement of number into columns and row. Suppose you have got this 1, 2, 5, minus 1, 0, 1, 10, 11, again like 5, like this. So when you are, you can multiply with, you know, multiplication of matrices are also possible. So when you multiply this, so what happens? The filter that, this is also, okay, let me put like this. This image patch is a square image, right? Square patch, right? The shape of the patch is square, like 5 by 5, 5 pixels by 5 pixels. Okay, so this is acting as a matrix. Now this filter is like 3 by 3 matrix, like 3 rows and 3 columns. And when it is sliding, so whatever the number is there in each box, right? It will be doing some calculations with its box, you know. So this box is over the top. Filter is on the top. Yes or no? Tell me.
Yes.
Filter is on the top, okay? Now here you can see this pink color pink color dashed square. So this is the filter now in this region. So here -1, 0 plus 1, minus 1, 0 plus 1, minus 1, 0 plus 1. Understood?
Yes,
टेबल
ma'am.
बाद में रख लेना, टेबल रुक जा, बाद में रख देंगे, अभी बैठ जा चुपचाप. So what happened? So minus 1, when it is coming on a box with the value 10, can you see over here? 10 into minus 1, multiplication is done. So the next
10.
box is this 10 into 0. So this 10 is for patch and this 0 is filter number. This 200 is, this 200 is your patch and plus 1 is your number assigned to the filter. You don't go deep into this, just whatever calculations are done here, okay, while sliding, you must get a higher output. Whenever you're getting a higher output, that edge is detected. So in layer 1, I told you, in layer 1, it is detecting vertical edges. Detecting vertical edges is very simple task. So when we converted this task into mathematics and we got a high number means yes, the edge is present. The probability of having a higher number, that means a vertical edge is present. If the number is low, for example, like 25, or your number is like 5, that means an edge is not present. Got it? Yes or no?
Yes.
Yes, ma'am.
Okay. So don't think, well, I should understand the calculation. Just understand whatever calculation is happening. If you're getting a high number, that means whatever it is trying to learn is present. Suppose in layer 2, okay, it is not trying to learn a vertical edge. In layer 2, suppose it is trying to learn if curves are present, if circular shapes are present. High number means curve is there. Circular shape means curves are there. Okay. Now, based on the colors also, we can try to find whether it is benign or malignant. More pink means benign, and if it's more purple or more blue, that means it is malignant. Okay. So for colors also, they can do certain matrix, element wise multiplication plus addition. So these are some things which you don't need to know the names. So how many layers? It depends on how deep you want that neural network. So never think the deeper the layers, the more better my model will be. Never. It depends, okay? Sometimes 32 layers can also work, okay? And not only layers, here we are talking actually about filters. In each layers also multiple filters can work. So here only one filter was there. One filter. Simultaneously, many filters can pass one by one. So one filter is for edge detection, other filter is for color detection. In each layer, many filters can be there. Understood? Yes?
Yes, ma'am.
Once again, I'll just tell you, layers are also there. Layer 1, 2, 3, 4, 5, whatever layers. In each layer, you will have filters. First filter will try to learn vertical edge. Second filter will try to learn curves. Third filter will maybe try to learn color. Second filter, whatever like this. So each filter can have, each layer can have multiple filters, and they can be, spread across all the different layers as well. So how many filters? It depends on your model. Perhaps 32 filter, perhaps 64, 128. And each filter will learn a different pattern. Got it? First two filters for horizontal and vertical edges. Filter 3 to 8, curves like that. Now, I told you that this filter will slide over. So there is one other term known as stride. How much it is hopping? Okay. So here can you see, I can maximize, I can't maximize enough. I hope it is clearly visible for you all. This diagram?
Yes, ma'am. It's visible. Yes.
Stride 1 means this is the initial position, and the second position is, it is shifting to the next column, just one place it is jumping. Okay, that is stride 1. Stride means how much you're hopping. So stride 2, that means at the whole window or the whole frame will jump twice, and the second window will reach over, here. This will be the second window. So you can set what is the stride also. So higher the number of stride, more faster you will get the result. Okay, got it? I thought that I'll be trying to. Okay, I have got 7 minutes left. Let me see whether I can teach you what is padding. So after stride, what you should know is padding. Please note down this. There is a formula, I guess, n minus f plus 1, I guess. Yeah. 5 plus 2. Just a second, let me confirm the mathematical formula. And if you know this mathematical formula, it will be easy for you to understand. Just a second. This
W
is the
minus
formula.
F plus
What
2.
formula I've written is correct. What? Sorry?
Yes, ma'am. N minus F plus 1.
Yes, it's n minus f1. Sometimes I get confused. Okay. So suppose, for example, I'm giving you an example. You have a 6 by 6 patch. Okay. So the patch is like how we took, 5 by 5 patch above. So we have got 6 by 6 patch. And in that, we are going to slide a filter, 3 by 3 filter. Okay. So the final output, the feature map that we are getting, we have to we have to calculate like this. So n value, this is the n value is 6 and f filter value is 3. So finally what we are getting, n minus f plus 1, it becomes 6 minus 3 plus 1. So your answer becomes 4. So the original patch was 6 by 6. You get it? But output that we are getting is 4 by 4. So can you see there is a data loss? If input
Yes,
was
ma'am.
6 by 6 patch, we must get a 6 by 6 output as well. Yes? Others?
Yes.
So this
Yes, ma'am.
formula will calculate the loss, okay? Now suppose it's not like suppose you don't want the data to be lost, okay? So what you do is, for example, if you're taking a 6 by 6 patch, you want the output also should be 6 by 6. Got it? You don't want any data loss. So what you do, mathematically calculate n minus f plus 1. You calculate this is equal 6. You know your filter value is 3. 1 is constant. So let us decide what should be the original value of n. Okay. So that even by giving the input as 6 by 6 patch, you are getting output as 6 by 6 patch itself. So answer will be like, if you solve, answer would be n equals 8. Okay, so what the input to get a 6 by 6 output patch, what you do, so you have got an original, I'm just making it 1, 2, 3, 4, 5, 6. Okay, so I've drawn a rough 6 by 6 patch over here. So, if you want an output as 6 by 6 patch as well, the value of n, that means the patch must be 8 by 8. Only then output will be 6 by 6. So the original input is 6 by 6 only, what you do is you add an extra boundary. You add an extra boundary, and this process is known as padding. Okay. So what you do, and you set the pixel value of these padding, extra padding, as 0. And what was your original input 6 by 6 patch now has become 8 by 8 patch. And since the extra patches have value 0, your original data is not manipulated. Got it? You have increased the 6 by 6 to 8 by 8, but you only had added 0. So your original data is not changed, and now you will get the output also as 6 by 6 feature map. So ma'am, we are just adding a 0. We are not changing the values. That's why we are getting the output. The conditions is satisfied. The n value is 8. That's why my final output will be 6. So to make n value 8, I'm adding an extra column and extra row. That is it. And I'm adding the number as 0. So this procedure is known as padding. Why have we done this? So the output data should not be lost. Got it?
Yes, ma'am.
Yes.
Okay. We have got only 1 minute left. There is one web page I've made, this one. I'll give you the link for this one, okay? By tomorrow only, I'll do all the pending work. This here. You see how the filters are moving here. So now, must be fast. So this is the filter, okay, here. So now it is on all 20, 20, 20, that it is on black patch. And you can see the output is 0. Now what you do, you click on next. Can you see the your filter sliding? So it was on previous and now the filter is sliding. And still it is on black patch. Yes?
Yes.
It's tried of 1 and the output is 0. That means, okay, this is not the, you have not encountered any edge because output is very less. Now, like this. Now you see you have got output as 600. You have encountered an edge. Now you got it? Now also you are on edge. Now the output is 0. Got it? I'll give you the
Yes.
links, okay? Now the time is over. You can leave the class for now.